Okay.
Okay.
So.
Okay. Email time over.
Everybody caught up? Thank you, Loes. So I'm Loes Olde Loohuis. I have a PhD in computer science. No, that was a joke because of the failed. But it's actually also the truth. Okay, so before I start, I have something to disclose, which is that yesterday I flew from Europe with a budget airline and multiple layovers. And I did this with 3 little children, one of which is like a giant 1.5-year-old with a character that technically, according to the airline, still qualifies as a lab infant. So you know, if you've ever experienced either of those two things and you try to combine them in your head, you can imagine how happy I was this morning when I could kind of step out of that mess and get here on my own in the car. But also, if I stop talking at some point or something like that happens, somebody can just gently let me get going again because I'm a little bit out of it. Okay. Everybody can hear me well? Okay, so today I'm going to talk about the transcriptome analysis in whole blood reveals increased microbial diversity in schizophrenia. Before I start, this project is joint work with Sergey, whom already presented yesterday. He was really my partner in crime in this project, and we did everything together. So everything that I'll present today is the result of our joint efforts. Okay. So until about 10 years ago, people used to think that the blood, if you don't have sepsis, the blood is sterile. And then recently, people have started to look into the existence of blood microbiome in healthy individuals. It's kind of a difficult journey because the blood microbiome is really hard or nearly impossible to culture because there's so little of it. It's thought to be sterile. So that's very hard. There are studies that try to characterize blood microbiome using targeted sequencing. But of course, the problem of targeted sequencing is that you only find what you're looking for. And so we were thinking a couple of years ago already by now that if there's microbial diversity, if there's microbial activity in the blood, and we have whole blood RNA-seq, can't we find RNA from these microbes in our regular RNA-seq data? And so that's the research question: can we detect microbiome in blood using unmapped RNA-seq? So that's what we set out to do. We had the following cohort. We had it available to us of 3 disease groups and controls. The disease groups were schizophrenia, bipolar disorder, ALS, and controls. While it's mostly practical consideration using this cohort, it is actually interesting to study psychiatric illness as well as ALS in the context of the microbiome, because for all of these diseases, some role has been hypothesized. Specifically for psychiatric illness, there's this notion of the gut-brain axis, that somehow the microbes in your gut affect your mind. But also for ALS, even more specifically, it's been thought that certain infections can cause ALS. So we had 48 samples, roughly, for each of these disease groups, and we set out to tackle this problem. So this is basically our pipeline. The big one? Yeah. Oh yeah. I'm so scared now because I'm afraid the same thing will happen. I already have all these technical problems. So, so we started with this cohort. We had pyrophil whole blood. We did RNA sequencing, regular paired-end RNA sequencing. This is ribo zero depleted RNA, and we aligned our reads to the human genome because we're not interested in the human genome. We use the unmapped reads as candidate microbial reads. But because, of course, unmapped reads, I'm sure Sergey talked to you about this a lot already yesterday when I was in my budget airplane. There's a lot of junk in your unmapped reads. That's where all the junk is. So you have to be really careful if you want to make inferences from this little bit of junk that you usually throw away. So what we did is we filtered out low quality and low complexity reads. We filtered out reads that turned out to be human after all if you just blast them. You know, sometimes the alignment is not perfect and all these things are somehow stochastic. So that's quite a large chunk. And what we were left with is high quality unmapped reads. And those are candidate microbial reads. And so we took those and we used them for microbial profiling. So microbial profiling we did using PhyloSift, which is a tool that uses the phylogenetic tree of the microbes and takes candidate reads and using specific genes that are nicely able to distinguish the phylogenetic tree, uses a likelihood approach to match those reads onto the phylogenetic tree, and so you can see if a certain taxa is detected or not. So using this approach, we were able to observe 1,800 taxa with 23 different ones at the phylum level. So here's your kingdom. Phylum level is pretty high, but we decided to do our analysis on the phylum level because there's little microbiome and we have most power. So mostly we observe bacteria and some archaea. So this is what we found. Every little bar is like an individual. These green ones are the proteobacteria. They are known to be most common. And these are other types of phyla. And so if you look at this, right, it looks like, oh yeah, schizophrenia has a lot going on, kind of. And that's also what we thought. But before we interpret these results, I want to go to this quote that some of you know already. As a researcher, you are the defender of the null hypothesis. And this may be a little bit naive quote. Eric Sobel is a statistical geneticist at UCLA, and it's kind of funny because he said, to imagine him saying it, but even if you're not at UCLA and you don't know Eric Sobel, it is still an interesting quote because I think us as researchers, young people trying to make it in science, we know we have to publish, right? And we know we are rewarded by working hard. We're rewarded by being, smart. We are rewarded by being creative, maybe. But we're mostly rewarded by getting results and publishing in whatever Nature and Nature Methods. And therefore, it's sometimes easy to not defend the null hypothesis. And it's just a digression, it's whatever. It's not really related much to the talk, but I think, we all know how to set up our simulation so that our method is a little bit better than the other method, because we know all the assumptions, and we know everything that we did. And so I really take this to heart. I think we are, there are no alternative facts. We are the ones to do this, and we are the ones to stand for our research. So that's what we did. Before interpreting these results, we really tried to scrutinize if this pipeline really makes sense, if what we find really makes sense. And so the first thing we did was a negative control experiment where we had 3 LCL cell lines. They are supposed to be sterile. And we were just applying these methods to see if we find microbiome. We used both PhyloSift and another method, Metaflan, and we found nothing, which was nice. The only thing that we did find was the Epstein-Barr virus, which is the virus that is used to transfect the LCL cell lines. So that was actually really nice negative control with a little bit of positive control. We also did another positive control experiment where we took RNA sequencing from epithelial cells that were infected with chlamydia to see if we could detect chlamydia. And we did. And in fact, we could even see kind of a dose-dependent effect. If they'd just been transfected, it was lower than if it was after 24 hours. Another thing that us and reviewers are worried about is skin contamination. A reason we are not so worried about it is that we have, for each individual, we have 2 tubes and we use them randomly. So if it's just from the, from the needle, at least half of the samples should not have this problem, and we don't see such a distribution. Then there's lots of technical and experimental confounders. The person who did it, the run, the batch, the site of the collection. We looked at all of these and we found no effect, no differences in terms of microbial composition. Finally, we also looked at whole exome sequencing rather than RNA sequencing for a couple of samples, and we found good concordance. We looked at existing literature, most notably the Human Microbiome Project, which has 500 samples and characterized using targeted sequencing those 500 samples in a gut, oral, airway, skin, and vagina. And we found that of the phyla that we discovered, 15 were also found in HMP, and 13 are confirmed by at least 10 samples. And also, our sample looks most closely related to oral and gut, which makes sense that in the way that we think about it. Whatever is blood microbiome comes most likely from your spillover from your gut or maybe oral cavities. Also, the few blood microbiome studies that do exist show similar abundance of the most abundant taxa. Okay, so back to this picture. Now it looks like there is more going on in schizophrenia than in the other groups. And we decided to formalize this using a notion of alpha diversity, which is very commonly used in the microbiome field, which basically looks at the relative abundance of different taxa. So you look at how many things you find and also how abundant they are relative to each other. So if you have one taxa that is like super abundant and a little bit of else, you don't have really high diversity. But if you have like kind of a balanced abundance, then your alpha is really high. And we do see that schizophrenia samples have a higher alpha diversity than the other groups. And we don't see any difference between the other groups. It looks like maybe controls are a little bit higher, but they're not statistically significantly different. P-values are based on ANCOVA, correcting for 6 tests, and we normalize our alpha values for all the technical confounders that we could think of, such as RIN, batch, flow, cell lane, etc. And the total variance explained by reduction in Nagelkerke R square is 5%. These results are not dependent on age and sex. Schizophrenia samples are a little bit younger, so we also did a subset analysis where we only look at older samples, younger samples for everybody, and we see the exact same result. They're not dependent on the number of unmet reads per sample, or the technician, or etc. Or the level of taxonomic classification. I said we do it on the phylum level, which is this one, but at the other levels, we see the same trend. So the next question is, okay, we see this increased microbial diversity in schizophrenia. Is it because schizophrenia samples have a specific microbial profile that is different from all the others? Or do they just have an increased microbial diversity in general? And this is also very commonly studied in the microbiology field, which is beta diversity, which basically looks at the diversity between 2 people. So if you have 2 people, you just compare you take the minimum of the relative abundance of each of these taxa. So if you have exactly the same, your beta diversity is 0. If you are completely different, it's high. And what we see, so if schizophrenia samples have a specific profile specific to schizophrenia somehow, you would expect that the diversity between two schizophrenia samples is lower than between schizophrenia and controls, or between controls and controls. And what we see is that to some extent, so what we see is that schizophrenia diversity is still higher than controls. Schizophrenia to controls is a little bit higher even in schizophrenia, but barely. So we think that most likely it's not due to specific microbial profile. This is the same if we include original diversity into our analysis. But we do find two taxa that we observe more frequently in schizophrenia cases compared to the other groups. And these are these two. Sometimes I try to pronounce, but now today I don't feel confident enough to pronounce these two names. But we do know that, so here you see number 1 is, we see it in 20 of the 48 schizophrenia cases compared to 5, 3, and 2, which is really a big difference. And these two are actually not correlated. So they're not the same schizophrenia samples that have these two. One thing is that the Planktomycetes is a group of gram-negative bacteria that is closely related to chlamydia. And we do know that chlamydia infection is very common in schizophrenia cases. Okay, so in addition to this reference-based method, we also use the de Bruin method. Actually, is David here? You're David, nice to meet you. I saw you on the program and I was like, oh yeah, that's great. So David did this work. So basically, I get a little bit nervous now that he's here. But basically, what the method does is you use your candidate microbial reads and you transform them into a de Bruin graph. And then you have two people, two de Bruin graphs, and you see what is how many changes do you have to make to get one de Bruin graph into another. And given those distances, you can do PCA on that. And so basically, these are the PCs from the de Bruin method, and these are the PCs from the phylosafe tree edit distance method. And we observe that, first of all, we observe that these schizophrenia cases are very different, but also the Bruin and phylosafe are in close concordance with each other. And they also, if you just measure the de Bruin distances, these are correlated with the beta diversity. So it's another way, without using references, to kind of study if there's certain group-based patterns. Okay, so as a final effort to defend the null hypothesis in this case, we attempted to do a replication study in an independent cohort to see if we see this increased diversity. And here, this is the original picture that I showed, and this is a replication where we have 88 controls and 92 schizophrenia cases. And again, we see an increased microbial diversity in schizophrenia. There are a few caveats to mention, or selling points, which is that in this method we use ribo0, and this method is poly-A-selected RNA-seq, so it's a little bit different. Here we use phylosafe, here we use metaflan because of the poly-A tails. And this that I show you here is at the genus level, whereas this is at the phylum level. But since this holds also at the genus level the same way, it's not really, I think, a problem. Okay, so what could this all mean for schizophrenia in terms of, what does it mean? So one thing is that there's this long-standing hypothesis that immune system plays a role in schizophrenia. either during development or later on or during adolescence. And so we wanted to study the relationship between microbial diversity and immune system in this cohort, but of course we didn't have data for that. But what we did have was DNA methylation of 65 controls in the replication cohort. And what you can do with DNA methylation, among other things, is that you can estimate cell type composition using the Hauserman method or the Horvath method. And so what we did is we looked at our microbial diversity and to see if there's any relation with cell type composition in this cohort. Anil, by the way, did this work, whom you just heard. These are all the cell types that we can estimate using the methylation data. And we looked just basically if their relative abundance is correlated with alpha diversity. And we find one of them is strongly negatively correlated with alpha diversity. And this is these p-values, even if we correct for all the tests, if we take the normalized levels of alpha after correcting for all the batch effects and stuff, as well as all the covariates that are common in methylation data. So there is something with the immune system. CD8 positive cells are differentiated T cells, and if you have fewer of them, you have more microbiome in your blood. Okay, so using our lost and found pipeline, we are able to detect microbiome, and we can do this in a robust way. And Sergey took this even a lot further by, completely dumpster diving everything out of it, not just microbiome, but what else is there in the unmet breeds. But he already talked about it. We do observe increased microbial diversity in schizophrenia, and we replicate this in an independent cohort, and we observe a negative correlation with CD8 positive T cells. So that's it. Still, there is a question: what does it mean? What are those microbes? Are they real microbes that go across the gut-blood barrier, or is it just the RNA that goes across? Can we measure gut permeability to see if that can be directly linked to alpha diversity? Is this a cause or is this a consequence of disease, or both? We looked at polygenic risk scores computed from the GWAS, and we found no correlation, but of course there's limited power there. What is the link with the immune system? And I already mentioned what else is there in the unmet breeds. So this is the last slide. Thank you to everybody, especially Sergey. Yeah, that's it.
Yeah, so we'll continue this final point. So often, things can be explained by differences in cell type composition.
Yeah.
Viewed as a confounder. So I mean, if it's the case that it's known that microbiome differs by different cell types, and it's known that cell type composition might be different in this phenotype, then does microbiome have an effect beyond cell type composition?
Yeah, I mean, so one thing I didn't make clear enough is this is only in controls. So within controls, we see this negative relation. And
But when you're doing
yeah,
analyses, were you correcting for the cell type composition?
no, not in the original analysis.
So the facts still hold if once you correct for the cell type composition?
Does anybody remember if we checked this? It's a very good point. I will check it. My guess is yes, because it's a stronger effect than this correlation. But it's definitely worth I mean, it's hard to check, actually, because we don't have this methylation data for the cases. So, yeah. Yeah.
So have you tried to use the RNA data itself to actually estimate the cell composition? So our lab has developed both methods, both for methylation and also for RNA sequencing. You can use the same exact data to get a signature to the cell and estimate that.
Yeah, no, we haven't. And that actually solves also that question. So that would be great to do that.
Yeah, if you want to talk later.
Yeah, I would love to talk. Yeah, yeah, I would love to check that out.
And I have another question in terms of the number of reads that you're using to establish these connections.
Mm-hmm.
How many unmapped reads are you starting with on average, and how many of those are actually being identified as a microbial read?
Yeah, good point. We actually normalized the number of unmet reads that we start with to 100,000, and we of those about 1% is identified as microbial. Yeah.
And when you look at some of these microbes, especially the ones that were represented in the schizophrenia, have you ever tried to map back onto those to see is it just how many reads are actually happening and do they actually, are they good alignments?
Yeah. Yeah.
Just these,
These specific marker genes. No, we haven't done that. That would be a really nice thing to check as well. Yeah, no, we haven't done that. That's a really good point. Thanks.
Do you have access to medication use data? Or does anybody know anything about how clozapine or antipsychotics affect microbiome?
I don't know. We should have that for the schizophrenia cases. Yeah, it would be really nice to check. And yeah, it would be really.
It could be very interesting if the increase in diversity might be pointing to that there's actually a lot of transition in the microbiome in these patients.
Yeah.
Modified medication or some other
Yeah.
thing. And that's oftentimes what you see with the microbiomes that are disrupted a lot.
If there is fluctuation. Yeah, that would be really nice to check. So in addition to checking just whatever medication they're on, also look at the temporal component there, you're saying?
I guess just you can look into as a kind of biological explanation
Mm-hmm.
to see if there's anything that may be causing more disruption
Yeah.
in the microbiome
Yeah.
of these patients versus the other controls.
Mm-hmm. Mm-hmm. Okay. Yeah, that's great.
So maybe related to this question. So did you develop, derive any significance check, any p-value for your result about the schizophrenia?
Yes. Oh, yeah. So for example, this p-value is 0.006 after correcting for multiple testing. For the original results, this was an ANCOVA with 6 tests. So these are the individual p-values, 0.0005, 0.0021, 0.0001, after correction already for these 6 tests. Is that your question?
I think so.
Mm-hmm.
Like significance of your bottom line theory.
Yes.
That, it's more diverse in schizophrenia.
Yeah.
So this is
Yeah, these are the p-values, and this one is 0.003.
And did not continue to any biological meaning of what you found?
Well,
Genes or microbes or anything?
well, so we found for these specific, and there is a little microbiome in the blood, so we couldn't really do so much more than looking at diversity or these notions that kind of aggregate what's there, to get most power. But we do see significant difference. And again, these are corrected p-values for these two, for the planktomycetes and the Termatogae. What that means, yeah. So our microbiome collaborator in the Netherlands thinks that this may have to do with the increased chlamydia infections in schizophrenia, because in previous studies it's been shown that 40% of schizophrenia patients have chlamydia compared to 7 in controls, and we see very similar ratios. And these are part of the same superphylum, so it might be that the phylocyst is like not really good at distinguishing them. But yeah, I mean, we would love to study this. We would love to study this further. We started off this project not even knowing if we could find, anything using this method. Yeah. David.
You say that schizophrenic patients are associated with increased diversity in the microbiome, but it's not clear which direction is causing it.
Exactly. The increased diversity is very clear, and 5% of the variants, we see that both in the original discovery sample as well in the replication. That is a big chunk. So it's a very strong effect. We can see it in small samples. But yeah, whatever. So for example, one of the things that we want to do is look at in follow-up studies, because we don't have it for these individuals, but look at markers of gut permeability and correlate that with, to see if it actually, if due to either medication or maybe genetic factors, the gut is more permeable, and therefore there's this hypothesis that there's a long-term stress on the immune system on schizophrenia patients, and therefore it kind of slows down, and you get this. But this is all hypothesizing. I don't know if any of this is, true.
So are there any additional microbiome studies on schizophrenia patients taking gut microbiome?
There is one study in gut microbiome. They see more microbiome. They see differences in microbiome, not necessarily more. It's a very small sample, though. It's like 20-something cases.
Whether the difference is already
in the gut already. Yeah.
small plate.
Yeah, if you see a little, yeah. But the thing is, with having a lot of diversity in your gut is generally thought to be a good thing, but having a lot of microbes in your blood is thought to be a bad thing. So Are
there other conditions or population which are known to have greater diversity of microbiome?
In the blood, nobody's ever studied it, as far as I know. But in the gut, there are studies with type 2 diabetes, where in the gut, so I repeat, if you have a lot of diversity in your gut, it's good for you, for healthy individuals. And so with type 2 diabetes, you see that these have a decreased diversity. That's one thing I can think of. So there are some studies out there more generally looking at the microbiome.
And if you look at the type 2 diabetes, do you think you'd also see it in the blood?
I don't know. I don't know. That would be nice to check. And there's lots of RNA-seq out there, so we could check.
So you mentioned something smaller and big question. So you mentioned something about Schizophrenia and some database. What exactly you mapped there? What is the 16X? What exactly you mapped in this database? What exactly?
That's an existing tool that's called Pvalues.
Yeah.
It uses genes that are present in a single copy in the genome that are representative of certain junctions in the phylogenetic tree. And it maps using maximum likelihood, it maps reads onto the tree and sees, and using a specific test, estimates whether, with a certain likelihood, that taxa at that level is present. So this particular library that we use uses 800 genes across. So these are curated genes at a single copy, etc. So that you don't have like noisy genes, and so that you are able to distinguish it from human genes as well as other microbes. Any other questions? Okay, well, let's play some soccer.